Just keep those.
Desktops. Hopefully Rose pops back in.
Yeah.
Having some problem.
If not, Brent will do the old switcheroo. And you
No
can go
problem.
first.
Yes, no problem. Is Rose in Australia now?
Yeah, yeah. Yes, you said she would be back in for the start. I'm still sharing my screen, aren't I? Yeah.
Yeah.
Right. Give it 1 minute. Okay, let's go ahead and get started. So it's really my pleasure to host this seminar for this month, the topic of which is species delimitation, conservation, and speciation. And we've got two really great speakers here. We've got Professor Rose Andrew and Professor Brent Emerson, who are going to tell us about this topic. For those that are not familiar with the format, we're going to have essentially two 20-minute talks with roughly 5 minutes of questions after each. And then following that, we're going to have 20 to 25 minutes of discussion around this general topic of species delimitation, conservation, and speciation. And before we get started, I will just plug the next seminar, which is on Tuesday, the 13th of May. The topic will be speciation and global change, 5 p.m. CET, so definitely join us for that. And with that, I'll get Rose to share her slides and I'll give her a short introduction. Great. So yeah, it's my pleasure today to introduce Dr. Rose Andrew. So Rose is an associate professor of plant molecular ecology at the University of New England. She has a diverse background in ecology, genetics, and the evolution of wild plants and animals as well. Rose did her PhD at the Australian National University, focusing on the ecological and quantitative genetics of chemical defenses in eucalyptics using population genetics, phytochemistry, and field experiments. She then went and did a postdoc at the University of British Columbia, where she used landscape genetics and population genomics to study the evolution of sunflower ecotypes in sand dune habitats. And in 2014, Rose started the Molecular Ecology Lab Group at the University of New England in New South Wales, and she's developed a research program focused primarily on speciation genomics in the genus Eucalyptus. And today she's going to tell us about adventures in eucalyptus, trying to make sense of species in apparently promiscuous trees. Thanks so much, Rose. Take it away.
Thanks a lot, Sean. I hope you can hear me okay. So I haven't followed the brief particularly well, and part of the reason for that is that I'm a bit of a born-again botanist. So I studied botany as a uni student, and then went away and did a whole bunch of fancy genomics. And then since I came back to Australia, I've become increasingly sucked into plant systematics and particularly species level plant systematics, which drags me away from the thing that really keeps me awake at night sometimes, which is eucalyptus speciation. So because it's the thing that I think about whenever I am not having to think about shrub species delimitation, I've gone a little bit back to thinking about species concepts and what makes sense for eucalyptus. So you can probably guess that PowerPoint's AI generated this beautiful slide for me, but I couldn't pass it by because it picked a beautiful pink flowered eucalypt, which is actually vaguely relevant to what I'm going to talk about. All right. Oh, hang on. Is it going to work? There we go. All right. So I'm going to talk a little bit about why I'm obsessed by eucalypts and what the problem is with species in eucalyptus. And then then have a look at some of the things that we've been doing to try and understand whether gene flow is really pervasive among close relatives of in eucalyptus. And then thinking about what does this mean for concepts of species in this group. So my interest was piqued in eucalyptus after doing honours in acacia. I started doing a PhD project looking at chemical ecology in eucalypts, and I was using microsats, and I was really fortunate to be able to borrow other people's microsats from other species, and they tended to work really well in the species I was working on. So that was the first hint that made me think there's a lot of shared genetic variation in this group. And then I started looking at the chemistry of the leaves and found that there were some really striking chemotypes. So this is a bar chart showing the relative peak area of some of the main chemicals. And this chemotype is the classic one. The compound that's dominant here is cineol, which is the main constituent of eucalyptus oil. But some of the individuals in a lot of populations had traces that had compositions that looked more like this. So they were dominated by a range of different compounds, and then some looked like this, which are really different. So this stood out to me, partly because in preparation for doing this part of my project, I went and visited some people in WA, and they were working on a completely unrelated species, but they had found that they had two chemotypes which really looked like this. So when I found them by surprise in the species I was focusing in, I started to think this is really interesting. Why is this shared? And then finally, eucalypts come in lots of different shapes and sizes, but the vast majority have cream or white flowers. So it's the colour's made up by the filaments, and in most of them, they are not this beautiful pink colour, but there's a group of closely related species that are all polymorphic for this flower colour difference, which I thought was really interesting. And then it kept in my mind when I went away and worked on sunflowers for a long time. So when I came back, I started trying to find ways to work on eucalyptus again. So eucalyptus has about 600 to 800 species, depending on who you believe. And it occurs in a huge range of habitats across the continent. The species range from really widespread to very narrowly distributed. And they're arranged they're about 60 to 70 million years old. But sorry, I've got to hide the faces. So they are grouped into subgenera, and that's what we're seeing in this phylogeny, labelled on the right. But within the subgenera, they're grouped into sections, and these are some of the major sections. And the sections are then grouped into series. And in this phylogeny, the colours represent the series. So you can see that these series, which have been defined by morphology, sometimes hold together, but often they really don't. in trees. So one of the key features here is that that there have been some really rapid radiations. So extreme shifts in diversification rate have been found in these very diverse groups. So Bisectae, Maidenaria, Adnataria, and Glandulosea. So Adnataria are my favourite ones, and most of the ones I'll be talking about are in that group. So hybridization and integration has been a hot topic in eucalypts for hundreds of years. So they were first recognised in at the start of the 19th century, but it was argued about by a lot of early figures who did a lot of important work here. But it was really nailed down by Lindsay Pryor, who did some beautiful studies with just morphology. So looking at, he found some trees that looked intermediate between these two species, and he collected seed from them and grew the putative hybrid offspring and found segregating variation in the offspring, which is really nice. It's kind of sobering to think that despite all of our fancy genomic technologies, some of this you could just do with morphology. And so this is a likely contributor to some of the muddy taxonomy that we see in eucalyptus. Anytime you don't really know how many species you've got within about 100 or so, it's a sign that people can't agree. Okay, so we can see that hybridization has occurred, but a really important review in 1988 looked quantitatively, so it's quite a long time ago, looked quantitatively at which species had known hybrids between them, and they determined which species possible hybridizers based on their distributions and whether they were reasonably closely related to one another. And they found that only 15% of those pairs actually had known natural hybrids in the wild. So it wasn't as rife as some had feared, but it was still, still a reasonable proportion of species that are hybridizing. One of the key take-homes that they found really was that the more broadly distributed species, so they counted the number of map grid cells and then compared that to the number of putative hybrids that were known. They found that there was an association between range size and number of hybrids being produced. Not too surprising, but that is potentially important. Okay, so more recently, there's been some nice work nailing down the rates of hybridization. So this was a study done in section Maidenaria by Matt Larkham, and he took a species within Maidenaria called Eucalyptus globulus, and he crossed it with a bunch of species both within the Maidenaria clade and between clades. And so the crossability was really quite high within that group, but much lower in other groups. So the percentage of hybrids that were produced in the I think these were the open pollen addition experiments that was shown in this particular figure. But yeah, so within Maidenaria, really high. Outside of Maidenaria, low, but definitely not absent. There's a lot more evidence for introgression in looking at the chloroplasts. So chloroplasts are typically maternally inherited in plants, and in eucalypts, there's, as far as we can tell, they are always maternally inherited. And so we tend to see stronger spatial structure in chloroplast DNA than in nuclear DNA. But when you look across species, and this was a study done by one of my former master's students who's just graduated with their PhD on a totally different topic, I was really excited to hear. Anyway, back to the topic at hand. So we sequenced chloroplast genomes from several species within one section of eucalyptus and built a tree and then plotted them on a map. And what you can see is that the clades in the chloroplast tree line up pretty well with geographic regions, regardless of whether they're found in the same species or not. So these haplogroups are shared among species and they're geographically structured. And even across series, which is another level of structure within sections, we can see some sharing of these haplogroups between them. So this type of pattern is what you'd expect if you have chloroplast capture or introgression. It's not a pattern that can be easily explained by incomplete lineage sorting, which is one of the other major explanations for shared variation. Because if you had incomplete lineage sorting, you'd expect the geographic patterns across species to be independent of one another. But instead, we see these clades that are geographically restricted. So this is a nice signal that tells us, yes, introgression has happened. So what do we think about potential species concepts in this group? It's pretty clear that the biological species concept, a strict interpretation of it, doesn't really work for this group. The barriers between closely related species are just too weak. We see lots of hybrids between them and lots of hybrids that are potentially fit as well. Ecological speciation, species concept, yes, we do see some niche separation. We do see that certain species, like rocky hilltops, for example, others can only live in fertile river valleys, but it's only partial. You still do see a lot of these species growing side by side. Evolutionary, who can say? We don't really know what the evolutionary trajectories of these species are. Morphological, phonetic species concepts, yeah, sure, but you often need multiple characters because a lot of the so-called diagnostic characters vary in space. Then we've got genic species concept. Unfortunately, we see relatively few fixed differences between some of these species, and we do see the potential that there's differential fitness between them. We do see some evidence of branch-specific divergence. For example, this is one of our favourite species, and we can see some peaks in the population branch statistic, which suggests that there's been some selection on these regions, with some caveats. The genotypic cluster concept actually is probably one of the ones that that makes the most sense, even though it doesn't have a strong mechanistic basis. Some of our species are parapatric and allopatric, so this is a real problem with the genotypic cluster concept, but there are many species that are broadly sympatric. So a lot of our work has been done in species that are broadly sympatric. And if we can't figure out species concepts that work, what about speciation processes? So one of the things that we do know is that these species have probably undergone range contractions and expansions through environmental fluctuations. We haven't had a large history of glaciation, but we have had fluctuating aridification. And another potentially important factor is that they have the widespread ones have very large effective population sizes. They have thousands upon thousands of individuals across the landscape. So one possible process that could have happened is that we've got ancestral polymorphism that might explain why variants are shared across species, and then we've had divergence in allopatry. And then the hybridization that we see now might be due to secondary contact. And you're probably guessing that I'm heading here because I've got the figure on the right. Combinatorial speciation is another possible process that could be really important here. So we've done a fair bit of work on this group of widespread Adnataria species that have quite strongly overlapping range distributions. And they're an important group partly because they're relied on by some highly endangered species like these beautiful birds. So some early work characterised the divergence and diversity. There's extremely high genetic diversity in this group and very low genome-wide interspecific differentiation. And intraspecific differentiation, which is this, it was even lower. Generally, we see that the series, which are groups of species within sections, tend to be distinct, and that's what we've got coloured here. But the species are not always distinct. We see some, and we see some putative hybrids here that have been borne out by the data, which is nice. So this got me studying and thinking about the spatial patterns of genetic diversity in these species. Within species, we typically see some isolation by distance. So you can see a latitude gradient in a principal coordinate analysis from Eucalyptus melliodora. And so it's weak, but it's pretty persistent in widespread eucalypts. We see it across the board. But then if we think about the spatial patterns of genetic distance across species, if we had no gene flow, we'd expect them to be spatially independent. But if we've got gene flow and isolation by distance, then we expect to see some genetic associations in space between those species. And so I thought back to an approach that Mike Whitlock thought up several years ago called the cross-species Mantell test. And this is essentially looking at genetic distance versus geographic distance, and then separating out the within species components from the between species components. So nothing too exciting, but it did allow us to test whether this spatial association across species was present. So in some species, we see no signal of it effectively. So these are essentially spatially independent, despite having overlapping distributions. And in others, we see reasonably strong patterns of association, which is indicative of gene flow or recent ongoing gene flow. So if you look at the patterns across the whole section, you see some interesting things. So one of the first things I wanted to do was to separate out the series. So I identified the comparisons in this data set that were within series. So these are close relatives of one another, and then between series. And then the cross-species Mantell test. Oh yeah, sorry. So some of these within series ones had really little signal. Others had relatively strong, significant differentiation between the species, but no strong signal of introgression between them. But for the between series comparisons, some of the highest associations in the cross-species Mantells surprisingly came in the species that had no spatial overlap, which is something that is kind of interesting and I want to follow up on. Then we had a bunch where there was a significant signal of introgression between overlapping species, a whole bunch that had no significant cross-species Mantell test, and one that was something else, and that I don't really want to talk about that one. That was a really unusual one that that I think comes down to perhaps an error in the species delimitation for one of those two species. Okay, so so this gives us a picture of introgression between some species, but certainly not across between all of the ones that have spatial overlap between them. So where are we at now? Ooh, whoops, sorry. Oh gosh, what's going on? Okay. Sorry, I'm going to have to scroll all the way through. Just jumped right to the end. Okay, so where are we at now? We're at the stage of trying to identify introgressed regions, the ones that are fixed and polymorphic. So we've used ELI for this, and we've got some preliminary results I've plotted onto the tree. There are some interesting things that happen. So what I've looked for is fixed regions of high probability of being introgressed between species. And so I've mapped the donor ones onto the recipient, and you see some interesting things. You see that there are some of the species that have really, really similar patterns, which to me suggests that maybe we're seeing some neutral signal that relates to incomplete lineage sorting and possibly things that happened before divergence. But we see some really interesting non-random looking patterns. For instance, this species, this light blue species here, has acted as a donor, but it doesn't seem to have acted as a recipient, which is quite interesting. And this also triggered some questions about whether these regions are associated with rearrangements. So that's the next question. Are they co-localized with genome rearrangements? And this rests on the work that Scott Ferguson did doing long read based assemblies of a whole lot of eucalypt species. And he found that there were huge numbers of rearrangements, many more than we expected, given the large-scale synteny across chromosomes. So combining that data set with our short read data, we were able to ask whether any of these groups of introgressed regions were associated with chromosomal rearrangements. It's really early days for this analysis, and I apologise for this horrible figure, but there were some interesting signals that suggested introgression in inversions or introgression in other rearranged regions, such as duplications or translocations. Sorry. Okay, so we're now starting to integrate this into landscape genomic studies as well, and it's too early days to share any results. But we've got some. I've got some more thoughts on this. It does look as though gene flow is ongoing, but it doesn't compromise most of the species currently. It also looks as though there has been quite a lot of historical gene flow, and the presence of these fixed introgressed regions suggests that it may have played a role in diversification. So combinatorial speciation is definitely a live possibility here. And then one of the ongoing questions is, what's the interplay of these processes with biogeographic history? Because of the long generation times, these trees live for hundreds of years, and they probably have a seed-to-seed generation time of about 70 years on average. So the number of generations since the last glacial maximum is actually relatively small. So that biogeographic history might really be playing an interesting role. All right, that's it. Other than my acknowledgements, lots of people have collaborated on all of this work, and yeah, wonderful folks.
Great.
Hey.
Thanks so much, Rose. Really appreciate that talk. There's going to be lots of interesting stuff to unpack in the discussion, I think, more generally about the topic. But any specific questions for Rose, feel free to throw them in the chat. But I'll just lead off with one. So you mentioned early on in the talk that range size seemed to be correlated with the incidence of hybridization in eucalypts. Could you unpack that a little bit more? Is that like primarily just about opportunities, like overlap with other species, or?
Yeah, I think of it as being about opportunities. One of the crucial caveats, though, is probably that the species level taxonomy for some of the very restricted ones is probably not all that good. There has been a tendency to see an isolated population that has some differences and describe it as a new species. And because it's an isolated one, it's not going to have known hybrids, but really it doesn't have any opportunity, and yeah, so I think it's a bit of both.
Yeah. Okay. Any other questions for Rose?
Yeah, early in the talk, Rose, you mentioned crossability, which I take to be kind of experimental hybridization. And then you talked about introgression. And what I'm just wondering is whether those two tie up. Is crossability a good predictor of introgression?
Yeah, and I put that figure in. It's not one that I wasn't involved in that project. It was pretty cool, but as I was putting it in, I thought, oh, I should really check exactly what how they defined crossability, because I'm not sure that they did it in the same way that that an Aquilegia study might have done it. It may be just something more like the proportion of species that they tested or something like that produced any offspring. So yeah, I'm not sure about that. And yeah, that relationship between introgression and crossability is, obviously not straightforward. You could have production of fit offspring that are infertile, and it stops there with no introgression.
Yeah, thanks.
Great. Time for one more question if anyone's got one. Brent's got one. Okay, great.
Yeah, I was just going to ask, in one of the PCAs you showed, I think it was Cytoroxon and another species, and there was a dot and a few dots in between, looked like just a bunch of F1s. So it made me think, you talked about like hybridization and fitness, but have you got any more information about the extent to which species produce hybrids and are viable back crosses, and the extent to which like that plot just suggested the F1s that it might be inviable?
Yeah, so that I meant to mention that in the field, whenever we saw something that looked like a possible hybrid, we targeted it just because we were curious. So we don't really have. I don't think we have good enough data to say how anything about the frequencies of back crosses.
Right.
Yeah, it would be really nice to do that, and we've got a couple of other projects on more range-restricted species that do seem to be intermixing with more widespread species, but it's we're getting some surprises in the results for that. So I'm not quite ready to talk about it yet. But yeah, really good question. Yeah, so that first step does seem to be happening in the field. Yeah.
Amazing. Yeah.
Yeah, thanks. Chromosome number variation. No, you didn't miss it, Natalia. Really appreciate the question. In eucalypts, they've all got the same chromosome number, and genetic mapping studies have found really strong synteny. So it was a real surprise to see, thousands of rearrangements in Scott's work. It really made us take a step back and think seriously about what's going on. And he's got several papers that have come out recently, and there's polymorphism in a lot of those regions within species as well. Yeah, it's very cool.
Great, thanks very much. I think we'll move on now to Brent's talk. Perfectly on time. If you'd like to share your slide, Brent. So it's a really great pleasure to introduce Brent Emerson. Brent did his PhD in Otago, New Zealand, with Graham Wallace, and then he moved to UEA, University of East Anglia in the UK, to work with Godfrey Hewitt, and stayed on there at UEA for a faculty position, lecturer and then reader, until he moved in 2011 to the Canary Islands, to Tenerife, where he's run a group ever since. Brent has really wide interests, an amazing range of organisms he's worked on, but there are some common themes, particularly in recent years, focusing on island biodiversity and its origins and maintenance and conservation. And also another common theme is very much the use of all kinds of genetically based methods, including barcoding and metagenomics, but also population genomics, to ask questions about diversity and its origin and maintenance. And today he's going to talk about the integration of barcoding and taxonomy through speciation theory. I'm really fascinated to hear how that's going to be achieved. So over to you, Brent.
Thanks. Thank you, Roger. So after the last talk, I feel very small. That was about a lot of genomic data, and I'm going to spend a lot of time talking about a very small part of a very small genome. I'd like to start by thanking Sean for the invitation, and for thanking Sean for some of his recent work on describing speciation as a continuum because it really helped me put some of these ideas together. So I'd like to break my talk down into 4 parts. First, just look at what is DNA barcoding and how is it applied for anyone who might not be familiar. Then look at why is there this apparent tension between barcoding and taxonomy itself. Then why is it useful to resolve the tension, and can speciation theory contribute to that resolution? So what is DNA barcoding and how is it applied? Well, it's, I guess, a bit more than 20 years ago, this paper came out, which has now been cited, I think, more than 17,000 times, which described the use of small regions of organelle DNA for the identification of species. So in plants, that would be the chloroplast, and in insects and other arthropods, it would be the mitochondrial genome. And then not long after this paper came out, which described how we can potentially use DNA barcode data, so mitochondrial data in arthropods, to discover new species, as well as assigning individuals to species. And this led to, the idea that in the addition of assigning specimens to known species, we also might be able to identify new species through DNA barcoding. And this is was a tension point, and that continues to be this idea of barcode-led species descriptions. But if we look at where we're at now, there we have a large amount of barcode data. It turned out to be an incredibly useful tool for a variety of questions. So on BOLD, we have, which is the central repository for barcode data, we now have more than 21 million specimens with a barcode sequence, and about 19 million of those are arthropods. So I'm going to focus this talk on arthropods. And if you look at BOLD data on arthropods, which I've summarized here, we can see that, well, we have across those almost 19 million specimens with barcode sequences, they represent 320 species, or 320,000 species. We also see this statistic here. There's almost 800,000 bins. So what are bins? Right, well, a bin is a barcode index number, and you can think of it as an OTU. It's a clustering of sequences. And sequences are clustered at around about 2.2% using an algorithm that's proprietary to the BOLD system. Okay, so OTUs are incredibly handy. They're a pragmatic way to group organisms, and then when we're talking about 21 million or 19 million sequences, it's a very pragmatic way to describe that variation. Okay. And it can also serve as a proxy for species, allowing for community level analyses. And when we talk about proxies, we're talking about numbers that should correlate with species richness, okay? And there's every reason to expect that there will be some correlation between OTUs and species and cells. Imperfect perhaps, but good enough. And so when we work with these OTUs, it allows community ecologists to really ask some pretty interesting questions at scales that were never really achievable prior. So here's an example from Rosemary Gillespie's group and Henry Kreinwinkel, where they've been able to use OTUs and barcode data to understand how communities assemble across volcanoes in the Hawaiian Islands. And this work by Paula Arribas, where by sampling across woodlands and grasslands, able to understand what structures arthropod diversity in soil and reveal that the turnover of communities occurs at very small spatial scales, indicating that evolutionary process is happening at very small scales across both habitat types. Right, so why is there then a tension between barcoding and taxonomy? Well, this paper came out in 2004, not long after the other papers I presented just before came out. And it described 2 tensions, if you like. One was this tension of DNA barcoding for diagnosing individuals, i.e. assigning an individual to a described species based on one mitochondrial sequence. Now, we now understand that there are limitations to interpretation, but it still serves as a valid tool. And it's just a question of being aware of the noise and errors that could conflate results. And then there's the second tension at the time was this idea of DNA barcoding for species discovery. And that's one that really, really exists through to the present. And this came from this study here, which identified or put forward a divergence threshold for birds above which mitochondrial variation probably indicates, new species. And so this tension, really, if you like, or this issue hasn't gone away completely. In fact, it's sort of been amplified of recent, okay, with several papers coming out recently that are describing species largely based on divergences in this barcode sequence region. Now, this is not a criticism of these papers. The issue is that with the vast amount of data we're generating, we do need a way to somehow move from all of these taxa that we're identifying that seem to have high divergences and formally describing them, because until we formally describe species, they are invisible to a lot of legislative bodies like public administrations and governments and what have you. So, this is a really valid initiative, but it's generated attention. And if we look at some of these species descriptions, we can see they're largely based on, the barcode sequence in the absence of other corroboratory data. Now, this sparked a large number of responses and unquiet, I guess you'd like, or discontent about the implications of moving forward with this approach, and how it may really do more damage than good to the taxonomic system. Okay, so across those papers that came out questioning this approach, well, the point was raised that there is, within these studies, there's no robust strategy for comparing against existing species that don't have a barcode sequence. So we might be redescribing already described species. That's the concern. That can, in practice, be got around now because of museomics. So we are having increasing success getting DNA sequence from collection material. And if we can get access to that material, then this issue can be overcome. But the second tension or issue that was raised was that, like, bins, these are OTUs, and they can serve as a proxy for species, but they are not by default species. Okay, so it's uncertain to the extent to which we should be using a divergence threshold to define new species. Okay, and so why is it then useful to try and resolve this tension? Well, there's a lot of really good reasons. Firstly, DNA sequence data is very taxonomically relevant. Okay, it can help us make progress with the Linnaean shortfall dramatically. So the Linnaean shortfall is the gap between the number of species that are formally described and the probable number of species that actually exist within a given taxonomic group. And it can also assist but not replace basic taxonomy, or not basic taxonomy, but classical taxonomy, which we refer to as a taxonomic impediment. That is the description of the fact that we just don't have enough trained taxonomists to deal with the number of taxa that have to be described. Okay, so I think there's what we need now is some way that taxonomy and barcoding can come together for a happy resolution, if you like, and we're heading in that direction, but there's still work to go. Another useful reason is that it's increasingly recognized that most species level diversity in arthropods is concentrated in taxonomically poorly known groups, which we refer to as dark taxa. These are groups in which we have very few species described, but we know that there's a lot of species diversity out there to be discovered and described. So the issue is that we have with these groups is that there are just too many species. They often differ by subtle or even cryptic species differences. We have too few taxonomists to process the material that would be required to attack these groups in a classical way. And we just have too little time because of these three features: too many species, difficult morphology, and too few taxonomists. Okay, and just a group that I can use to represent that issue is our work that we did several years ago, well, quite a few years ago now, with Francesco Cisonati, where we looked at like one of these classical dark taxa, Colembula, and we just looked at 7 species within one genus across these sampling sites in the northwestern Mediterranean. And in fact, most of these sites were sampled for 3 species. The other 4 were quite rare. So it's really a 3 species study. Okay, so we sampled these and we sequenced them. And this is the mitochondrial tree we got back from that data. And the mitochondrial tree is kind of corroborated by nuclear sequence data as well, but I'm just simplifying here. So if we just look at this line here, represents using a super conservative estimation of molecular rate, like a very slow rate. We know it's too slow for Colembula, but it estimates that at this point here, which is the Messinian salinity crisis, almost 6 million years ago, that we had in existence 34 lineages. If we use classical approaches to define clusters of sequences which might approximate species, we have 90, I think it's 93. So somewhere between 93 and 34 species. And these are all cryptic. I mean, these are all morphologically, I mean, we had a good trained taxonomist identify all these specimens. So these, and we actually know that they are species because we found some in sympatry and could look at co-segregation of nuclear markers. So this is the challenge that we have. How do you deal with this? Okay. How do you deal with this when you know that we just sampled in a very small part of the distribution of one of those species? This is its natural distribution across Europe. So this is like, the scale of the problem is phenomenal. Okay. Another issue is that these truly cryptic species can only be diagnosed based on molecular data. So we go back here. Now, most of the species level diversity that they're describing here is only diagnosable with molecular data. These are truly cryptic elements. So there's a definite role for molecular data in taxonomy. The other reason why it's useful is the generation of barcode data is increasingly scalable, largely thanks to recent time and cost efficiencies. So here's 3 papers that provide examples of how people are able to leverage ONT technology and MinION barcoding to generate hundreds of thousands of sequences at a cost of a single MinION run. Okay, so this means more people have access to generating data and more people have access to the data that's being generated. And the question now is like, how do we actually use that? How do we really deal with this like onslaught of data that we're going to be seeing in the coming years? Okay. So, yeah, where am I? Yeah, some reason my slides aren't moving. Oh, yes, they are. Good. Great. So how can speciation theory contribute to a resolution? Not provide a resolution, just contribute to it. Help us advance towards some sort of harmonious system. Well, if we look at how speciation theory is currently treated, it's not really. So despite this increasing focus on species delimitation, working species concepts are typically vague or not even defined at all within studies that are, contributing towards species delineation. I guess barcoding is conceptually similar to the phylogenetic species concept, but it has a threshold. So it's not really, it's similar, but it's not. But it is discussed in the context of the biological species concept. I've just put one example of that here, where describing discordance, that means where you have species that are actually clustered into more than one OTU. That it's been suggested these may involve reproductively isolated lineages that are not currently recognized as different species. That most of these splits, if you like, likely involve overlooked species. So we've got a reference to the biological species concept, and I think that's an incredibly helpful concept with the kind of data and sampling that we're typically seeing. So let's now think of speciation as a continuum, and this is where, like, you know, Sean's recent work really has been quite helpful for me. So, you know, we can think of speciation as having a gray zone, you know, between which reproductive isolation amongst lineages, you know, 0, until which it's complete isolation, full reproductive isolation equals 1. And speciation itself may be a discrete event. It may happen very quickly over a short interval of time, or it may be a very prolonged process. So we can summarize that here, you know, just sort of like really crudely. The time for speciation can vary greatly across different species group, from almost instantaneous towards very long and protracted. From the time points from which we get to our reproductive isolation is 0 to its 1 among lineages associated with mitochondrial DNA. Okay, if we look at a threshold for speciation, well, firstly, this whole notion of a threshold is kind of based on biased sampling data, where it's like taxonomically and geographically restricted. So some recent work, for example, you know, quite phenomenal studies sampling beetle species across Germany and neighboring countries. We looked at 3,500 species. We now see that, you know, up to 20% of species have interest-specific variation that largely exceeds the threshold that people are implying, and it can be up to 8%. And I guess it becomes a little bit circular. You know, you can ask, well, to what extent are they maybe looking at cryptic species? That may be a feature that explains some of this, but not all of it. The point being that a single threshold really doesn't capture reality. Okay, so you know, and it's basically inconsistent with what we know about speciation as a process. You know, this threshold kind of assumes that the time for speciation is, you know, there's a fixed time point at which speciation finishes. Okay. Based on mitochondrial DNA. So, using this singular value of mitochondrial divergence to demarcate species, you know, ignores what we know about how speciation occurs. But this incompatibility could be we could get beyond it if we start thinking about, you know, move away from this binary view of the speciation question. Okay, so so what we can potentially do is start evaluating, like, when does speciation complete, and in the context of lineages defined by mitochondrial divergence. And a straightforward approach to doing that in terms of the biological species concept is to focus on those lineages where we can find them in sympatry. In sympatry, we can sample individuals that belong to both. And using nuclear SNP data, which is now like it's largely become a non-trivial exercise to generate this kind of data. It's cheap and efficient, and for the purpose of what we're looking for here, it doesn't need to be, you know, it doesn't need to be an expensive process. So if we find in a generating SNP data whether we find these individuals largely do not segregate in principal component space, but with regards to their mitochondrial variation, well, that would indicate reproductive isolation between these lineages is 0. And if it's highly separated, okay, in that case, you know, there is reproductive isolation associated with mitochondrial lineages. And we've done that here in the context of the single species of Staphylinid beetle in the Canary Islands, where we found these huge divergences occurring within what's based on extensive morphological work, you know, cryptic. Everything does look the same. And we found 2 of these lineages in sympatry, so we applied this approach. Yeah, oh wow, the nuclear SNPs clearly differentiate these 2 things, and so that even with ancestry-based analyses, you know, we can apply those as well. And it's, you know, it's fairly clear that these things are not recombining in sympatry. They are good species, and that tells us that at this point here, reproductive isolation is 1, and we can by inference move back towards the root and assume anything further back is likely to be a good biological species as well. It's not, obviously it's inference, but we're basing it upon good quantitative data rather than subjectivity. So, what we can do is like look at different levels of divergence across different taxa and establish, thresholds above which things are probably belong to the same species and below which they belong to the same species and above which we're probably looking at different species. And by sampling across a fine scale range of thresholds, we can hopefully, or we should, be able to tighten up this gray zone that we're referring to, which enables us to really understand where we should be focusing our attention. By that I mean anything within that gray area is we can't equivocally associate it to one or two species. Okay. So this, gray area is conceptually related to the gray zone of speciation. So if we just take this hypothetical example, we've got a scarab beetle. I've chosen this name because it was the first species named by Linnaeus. Okay, suppose we have one species that we know in this group, Scarabaeus hercules. Okay, anything that's to the left of the gray area, we can reasonably assume that that is a new species. Anything below it, we can reasonably assume it belongs to the same species. And then to deal with anything that's divergences within this area, we can apply this qualifier that's been put forward that really describes that species status is unknown until further data can be accessed. Okay, so with this qualifier stat just recognizes that we're not really sure whether it belongs to the same or a different species until we check it out further. Okay, so it's identifying those taxa that really specifically need more work. And so, so how can this then be leveraged? Well, I'll just point out this recent study that's come out, which is on forward flies of Sweden. So this is an amazing piece of work where they, forwards are an incredibly complicated group. They're a classic dark taxa group. So in this study, they integrated barcodes with morphology. So typically 18,000 species, no one wants to go through and look at all of those. So their approach was to sequence everything first. And they can do this because we can scale up this barcode sequencing and it's cheap. So they sequenced everything, defined OTUs based on 3%, and then strategically looked within those OTUs at a subsample of individuals for morphology. Okay, they had a strategy for that, which makes a lot of sense. And so they just looked at 5% of individuals for morphology. Okay, and so they've got this 3% threshold that they're applying here. And what they found, oopsie, okay. And so what they're doing is accounting for morphologically diagnosable species below a clustering threshold. So that's super useful. But it can't really account for cryptic species below this threshold because it's based on, reassessing things that have differing morphologies. And it has less focus on species significance of divergences above this threshold. But if we look at this data, there's a lot of sympatry here. There's 315 OTUs from only 36 sites. So it can be used to describe a divergence range with the association within which the association between OTUs and species requires higher scrutiny. So it can help us to further concentrate our efforts. And this is, phenomenal because once we have these thresholds, forwards, okay, here we have 315 OTUs. There's just in one genus, the Megacelia, there's 1,700 species described, and it's estimated that in this one genus, there may be 170,000 species globally. So if anyone wants to attack the forwards in a different geographic region, we can apply these thresholds to help us focus our interests. Okay, I've gone slightly over. I think I'll just bring up my last slide now and thank the people who have funded this. A lot of this has been helped by being part of this Biodiversity Genomics Europe project, where we're dealing with massive amounts of barcode data and trying to understand how best we can use it. It's also been funded by the one of the Spanish ministries that funds science research. Okay, thank you very much.
Thank you very much, Brent. That's a very stimulating talk. I'm sure it's going to raise lots of issues for the general discussion, but if anyone has any specific questions for Brent, please pop them in the chat now. And I will maybe just kick off with one specific thing. I mean, using alternative methods to try and define where the threshold should be in a particular group is clearly helpful, and having the grey zone is an interesting idea. But I wonder whether we expect consistency in where that threshold should be across taxonomic groups, or whether actually we expect it to be, more similar in common species than it is and different in rare species, or something like that. Do we expect kind of taxonomic consistency in that?
Yeah, no, that's a good question. I think if we do it, we'll get the answer to that.
Yes.
Yeah. I mean, I wonder myself, well, maybe there is like a fairly universal set of thresholds that can be applied across all different arthropod groups. There will always be exceptions. Things will always fall out of those. So I think once we define these thresholds, they're going to be like, we can be 95% certain that's a species. You can never rule out the fact that, even more divergent lineages might might belong to the same species. But broadly, where those limits are across different taxonomic groups, and how different features of taxa, such as effective population size or whatever might influence that, is clearly super interesting. I think there's probably a lot of DNA, because we have all of this data in bold, and a lot of the DNA extracts are probably housed there as well. There's probably a really cool project could just be leveraged from existing sample data there.
Yeah. Thank you. So there aren't any other specific questions. I'll switch off the recording now, and then we can move on. Rose, did you have something specific you wanted to?
Yeah,
Yeah,
that is okay. Yeah, that was a really fascinating talk, Brent. It was really cool to see that. I love the idea of like interrogating it with SNPs to look closer. I just had a question about the process of using a sequence to diagnose a species. How often is there a holotype specimen in addition, or is it sometimes a photo?
You mean for what? For the species diagnosis? I think for those papers that I presented, there are holotype photos and there are paratype photos. Sometimes there's useful host data. I mean, in the case of those published papers, they were parasitic wasps. But that, I think the major criticism is that there's it's not an exhaustive morphological treatment either. I mean, there's pluses and minuses for what they've done. It's imperfect, but it's something needs to happen. So it's a step forward in many ways. Yeah.
Yeah, it's a cool way of approaching the species as a hypothesis, and it's the best hypothesis that fits the data that we have currently.
Yeah. I think the problem, another problem is that people treat morphological species as not hypotheses. They are basically what you should be matching against. And so there's this whole, I think it's really helpful when we start thinking about, yes, everything's a hypothesis. And in fact, we can start confirming or not hypotheses with, this, when we have these situations in sympatry.
Yeah. Thanks.
I'll stop.